anything and everything you wanted to know about genetic mutations and pedigrees. So, what is a mutation anyway? Well, that's any change in the DNA, and it has to be the order of like the nucleotide base sequence, the letter sequence. So, anything that is a change in what we're considered the norm or the normal DNA sequence. And this can occur really in any cell of the body. It can occur in your somatic cells and your germ cells. Now, somatic cells are normal body cells, basically anything that can go through mitosis. Whoops. And mitosis, remember, those are cells that they divide, normal somatic cells, and they are the ones that when their cell cycle becomes uncontrolled, they have uncontrolled division, and that causes cancer. Germ cells, those are the ones that pass on your lineage. We also call those gametes, and they are passed through the process of meiosis. And in meiosis, during the formation of those gametes, things can not always go as planned, and we can create mutations in offspring. Now, mutations, they can be caused by many reasons. Normally, they're just a mistake during DNA replication as DNA is duplicating itself. It can also be a mistake in mitosis or meiosis, and more oftentimes the outcome, which is protein synthesis, is where the problem lies. Now, there are certain causes which we call mutagens. Those are chemicals, they're in the environment, and they can cause DNA mutations. For example, UV light from the sun, that's a mutagen. Any sort of radiation, other chemicals like cigarette smoke and the poisons that they contain, those are called mutagens. They assist the DNA replication process to where it's more likely to form an error. Now, are mutations always bad? Not necessarily. I mean, sometimes a mutation can occur and it can really benefit an organism. Plus, mutations are how organisms get their genetic diversity. This is what makes evolution possible, and diversity can be a huge potential benefit to any organism. So there are two main types of mutations. There are gene mutations and then what we call chromosome mutations. Now, the gene mutation, that happens during DNA replication, and it causes a change in the original DNA sequence. There's something, usually like anything that's a change in the original sequence, like dwarfism or sickle cell anemia, which is just one nucleotide base, or dwarfism, which is a small series of genes. That's a gene mutation. Now, sometimes we can have a overall chromosome mutation. This is pretty much occurs during meiosis, or inside of the gamete cells, and that will change in the number of chromosomes, or like the overall location of the actual gene. So this is a very large scale type of thing here, where we're like losing or gaining a whole entire chromosome. Things like Down syndrome or Klinefelter syndrome or Turner syndrome are just examples of these large type of chromosomal mutations. Now, when we're talking about gene mutations, we can have a couple of things. We can have what we call a point mutation. That is just one nucleotide that can actually change. So you can have a whole sequence of things, like, for example, in sickle cell anemia, which is affects the red blood cells, there is a codon GAG, and then what happens is that A is replaced by U, and it's making GUG, and that one point mutation, that one single letter, basically causes the red blood cell to completely change its shape and giving the person sickle cell anemia. And when this replication error occurs over and over and over again, it produces an illness. Now, frame shift mutations, that's when we have a letter or two or three, these nucleotides that are actually inserted or deleted from an entire section or an entire nucleotide. So an example here would be if I have ATTACC, and then my new gene sequence here would be ATACC. We can see that this T right here has been removed. That's called a deletion. And my next example here where I have ATTACC, but I now have ACTTACC. This C right here, that is a new addition. We call that an insertion. So cytosine has been inserted into this gene sequence. And this basically like stops everything from being read correctly, and now it's thrown off even though it's only one nucleotide. But it basically affects all of the codons from here on out. So a one insertion or one deletion throws off the way that every codon sequence is read, and it can throw the whole protein synthesis process off. Now on that larger scale where we have an entire chromosome mutation, we have a couple of things. We have a duplication, a translocation, and a non-disjunction. A duplication, if you're looking at this part right here, this is a change in the overall size of the chromosome. It results in multiple copies of a single gene that are just getting replicated over and over and over again. So if we have a gene that's on a chromosome and during our process of where we're duplicating these genes, it duplicates itself over again, hence we call that a duplication. This is a large amount of DNA that is being read twice on a chromosome. We also have a translocation, and now this is a little weird here, but that's when a piece of some non-homologous chromosome, it's a chromosome that isn't really supposed to match up or is not related to that particular chromosome. But anyway, it's like a piece of a chromosome from some other chromosome is basically exchanged during crossing over, when things are lining up during metaphase. You will just exchange pieces of itself. So in my example here, we'll just say like this is chromosome number 1 and this is number 20. All right, well during when it's lining up in order to when it's going through duplication, this gene right here can just be translocated into another part of a gene, and now we have a gene that's on the incorrect chromosome. It's on a different chromosome. It's still there, but it's been translocated to someone who's non-homologous and someone who doesn't match itself. Non-disjunction, that's when chromosomes do not separate correctly during anaphase, and that results in like 1 or even 3 chromosomes rather than 2 per cell. So here in my non-disjunction example, this is my duplicated DNA cell, right, where this one's going to divide in half of it's going to go there, the other half is going to go here, and now we have chromosomes that are supposed to split up. So we're supposed to get an orange one, and a blue one, and a blue one. But in this case over here, we have our blue ones. Both of these guys get into one particular gamete where this guy goes here and he can split up. So this is what we call a non-disjunction. It's a whole nother chromosome that's been moved to one of the gametes, and this results in what we call unbalanced gametes. So one of the most popular forms of non-disjunction is Down syndrome. We basically have 3 copies of chromosome number 21. That's why it's called trisomy 21 inside of that egg or sperm cell. So chromosome number 21 it's really small, and so what will happen is that little pieces of DNA are more likely to get lost during the replication process than really large pieces of DNA. And the smaller the piece of DNA that's in those cells that is extra, the more tolerated it is. So on chromosome number 21, there'll be like a little piece, and you'll have an extra chromosome number 21 that will result in Down syndrome.